Welcome to Module 3, where we'll explore some basic but essential techniques and equipment utilized in medical laboratories. A good understanding of these fundamentals is the foundation for all other areas of testing. Are you ready to learn? Let's begin. 1. Microscopy and specimen preparation. One of the oldest and most important tools in any lab is the microscope. Several types are commonly used. Let's review. Compound light microscope. Most common design uses lenses to magnify 100. Applications include blood smears, urine sediments, tissue examination. Phase contrast microscopizes interferometry to enhance contrast of transparent or living specimens. For bacteria, parasitology, semen analysis. Fluorescence microscope detects fluorescent dye-stained structures when illuminated by specific light wavelengths. Vital for research like immunofluorescence and molecular techniques. The best microscope type to identify each sample. Tissue biopsy specimen. Compound blood film compound stool sample for ova or parasites. Phase contrast and cytohybridization. Slide fluorescence. Correct. You've matched the appropriate microscope to each application. Microscopy is foundational, but also still very relevant today. Proper specimen collection and preparation is also vital for accurate microscopic analysis and diagnosis. Let's look at some key techniques. Blood smears. Gently place a small drop of well-mixed blood at a 30 angle on a slide and spread with another slitty or dry and perform staining protocols as needed. Urine sediment exam. Centrifuge urine sample to concentrate cellular and cast sediments. Respin the sediment for microscopic field examination. Tissue sections. Fixation, processing, embedding, microtomy and specialized stains allow studying cell or tissue architecture. The incorrect statement about specimen prep. Blood is applied to both sides of the slide when making a smear. Urine should be centrifuged while still fresh, within 30 minutes of collection. Formalin is commonly used as a tissue fixative before sectioning. Stool samples need no preparation for ova and parasites exam. You picked correctly. Stool samples actually require various concentration and staining techniques before examination. Great understanding of foundational skills. I realize specimen prep may seem simple, but being detail-oriented is critical for reliability. Got any other specimen-related questions? 2. Laboratory equipment and basic principles. Let's move on to discuss some common equipment found across all areas of clinical laboratory testing. Centrifuge rapidly spins samples to separate components by density like cells from plasma or urine. Sedimentrators and speeds vary based on sample type and desired separation. Pipeteridite precise sample or reagent volumes. Mechanical or electronic models in various fixed or variable size. Proper technique is important for accuracy. Tubes and vials. Primary containers for collecting, transporting, storing and analyzing samples. Different materials for various analyses. Glass, plastic, etc. Spectrophotometry is how strongly a chemical species or complex absorbs light at a particular wavelength. Important tool for analyzing chromogens and colorimetric assays. Which sample type would require high-speed centrifugation? The student selects platelet-rich plasma preparation. Correct. Preparing PARP from whole blood requires very high speeds up to 3000 rpm to effectively separate platelets from other blood components. Well done. Basic concepts. A good grasp of some fundamental scientific principles is essential for understanding many laboratory techniques and test methodologies. A few important ones are Beer-Lambert law, absorbance and concentration relationships in spectrophotometry, enzyme kinetics, factors that impact reaction rates and enzyme activity, assays, reactions, stoichiometry, molar relationships in chemical or biochemical reactions. These come into play a lot in clinical chemistry, immunology, and more. Some additional details about microscopy and laboratory equipment. In addition to the main microscope types, variations exist, like the phase contrast inverted microscope used for cell culture work. Specialized microscopes have features like polarized light filters, dark field optics, differential interface, contrast, etc., depending on application needs. Advanced microscopy now involves techniques like fluorescence recovery after photobleaching, FRAP, and live cell imaging for research. Micropipettes are commonly used for manipulating or moving very small sample or reagent volumes less than 1 microliter under a microscope. Benchtop centrifuges are general-purpose models found in most labs, while high-speed refrigerated versions are needed for specialized applications. Graduated volumetric equipment like burettes, Volumetric flasks and cylinders deliver precise reagent volumes for standardizing solutions. Common spectrophotometers are single-beam models for microplates, cuvette samples, and continuous flow analyzers. Higher-end instruments offer features like dual wavelengths, kinetics mode, etc. Hematology analyzers integrate components like flow cells, hydrodynamic focusing, impedance technologies for automated cell analysis.